My name is Karan and I'm a student in Western Sydney University. Today I'm going to be presenting my assignment 3 for the subject of genomic data science, where I would be doing some analysis of, analysis of microbiome diversity in various kinds of objects that we're going to be talking later. So, what exactly is diversity? So diversity is one of the most widely studied ecology properties, and that is the most important to the field of ecology itself. So, what is microbial diversity? It is again a widely studied property from with the help of which various bacteria can survive if they are diverse enough. This is their defense mechanism, and this is how they grow. So we're going to be doing an analysis on those samples that we're going to be talking about later, and doing some analysis on that and discussing that. So as we're going to be checking the hypothesis for it, so let's say our null hypothesis is that bacteria is present on the objects, like on the objects that were sampled, basically, and H1 is the hypothesis contrary to that it is not present. So, but it's just a side note that it's not necessary that more the diversity or more the diverse an organism is, it's always good. It's not the case, but that's a completely different topic altogether. So next, so I'm going to be talking about just a little bit to the research so far that has been done on this. The majority of the research so far has been done into the patterns of diversity, but towards plants, and not this kind of research that I'll be doing. So, uh, it's still in its infancy. We can say that. Okay, so I'm going to be talking about the data set now. So the data set was collected by 5 people uh, that have 5% belongings that they would be sampling with the cotton swab. So collectively we collected 23 samples, and out of which there were 3 extra for negative control. uh, while doing the process, there must be some problem, or while collecting the data set or the samples, there could be a number of reasons for it. So but therefore the 5 item samples failed to produce data, and we have one sample also for the negative control. So basically, in our dataset, there are two more most important files. One is the sequencing file, and the other one is the mapping file. This is the tool that we'll be using. The below is the command for it. We're going to be using Chime. I'm going to be talking about this later. So we're going to be using different functions, tailored to it. But about the dataset, we're going to be just working with two main files. Uh, so Chimey is a tool that is used for genetic biodiversity analysis, and it's in the trend. It's very easy and free to use. So what exactly it is? It is a tool. I mean, first of all, it is pronounced as a quantitative insight into the microbial ecology. So it is a tailor-made thing for this particular purpose. So basically, in layman's term, what it does is it is a bioinformatic pipeline. That's what it is called, in which you just input the genetic sequence, and then it does its magic, and you get the outputs. And the output can be various things that you desire, such as some statistical outputs, some modeling, or phylogenetic trees, stuff like that. So it's a pretty cutting edge technology, and we're going to be using this. Next, demultiplexing. Well, demultiplexing is a process in which the original channels, the dataset that we create after sequencing, are extracted on the receiving end. So, but we're not going to be going into demultiplexing, since our data is already demultiplexed. So we are not gonna go into detail about this. But we still gonna discuss a little bit about demultiplexing. So this is the demultiplex result for of my data set. It is just to check the quality of our data, that at which sequence base it starts to deteriorate, or where should I take my sample till. So it is an quality plot, and that is obtained from random sampling, and the replacement, we haven't done it in a way, like it is without replacement. So this is a side by side comparison, as we can see. that in forward reads, the quality really starts to go below after like 230 maybe or 240, and in reverse it is somewhere around 180. So we can say, the data set starts to deteriorate in quality somewhere between 180 to, I'd say, 220 maybe. Okay, so we're going to be using a Fred scale for that to check it. Okay. So next is the summary that we get after demultiplexing. So it is a comparison of frequency and number of sequences, as you can see. Just the most at like 300,000, and here we can get the summary, basically what is the minimum, what is the maximum, the mean, the median, so stuff like that. This is the steps that generally is followed in the multiplexing techniques. So the data is filtered, the errors are learned, then goes forward reads, then reverse reads, and then there is some denoising. In this case, I have used the method pooled, and then you get the output. So this is basically the summary of number of samples with the number of features and its frequency. Now this is a graph and some details about the frequency per sample. It is it, it it is the comparison between the frequency per sample to the number of samples. So as we can see that, the median frequency is somewhere around 536. That is where it is settled down. Till that point we can be sure about our data set. It's a good practice, so we can take a range of the sampling from somewhere around maybe 500 to 600, but since I have the median frequency, so let's just go to the point. So I'm going to be taking 536 samples further for my analysis. So basically it is just the richness of the sample that is portrayed. The richness starts to deteriorate, so the quality. This is again the comparison of frequencies with the number of features and the frequency per feature. And here again we can see the median frequency as to 5 and all of the details about it. The next I'm going to be talking about the sampling details. So I have a side-by-side comparison of the item type without sampling, that is, without replacement. Without sampling. So left is without sampling, and the other one is when I've sampled to 536 according to my median frequency. So if I there's a clear difference between without sampling and the sampled mean frequency, as you can see, and there is a comparison of number of samples with the object that was sampled. So it is the item type. The next I have compared again with the 536 samples to without sampling, and it is done on the person data set. So there's a direct comparison. The next I'm going to be talking about the biological sex. Again, this is the comparison of without sampling and sampling to its median frequency that we have gotten from after doing the analysis. So, and this is again the sampling details when we talk about the ethnicities without sampling and then with the sampling. Okay. Alright. So next I'm going to be talking about side-by-side comparison of Texa bar plots. After I did my analysis, I used Texa bar plots to, visually compare and see the results that I've gotten. So basically I've talked about the indexes, item type, person, sex, and ethnicities in the box plots, and you can see, the results that negative control is the empty, and that means it works. So this is the provenance graph that it follows. So basically this is a graphical representation of the background of what is happening, where it is taking the multiplex data from, how is it sequencing, what's the next step, what goes where. So this is a general breakdown, or you can say of the process followed that is done by the computer in the background. So this is for the Texa bar plot. So I'm going to be talking about a different representation of alpha rare fraction plot. That's a function that was used for it. So and this is a direct comparison of the item type. As you can see that there is a direct flat line somewhere after like 300 with the number of samples and sequencing depth, and again the comparison comparison of depth with observed. So this is for the item type, and again this is for the person direct comparison of alpha refraction plot. So the color represents the person, and the graph is it shows the relationship between sequencing depth and the observed values for it for different people. This is for the sexes. There are only two, so we're getting only two graphs. So again, this is an alpha refraction comparison of the two, and this is for their ethnicities. Well, these are just the plots, but in the background this is what it's exactly doing. In the background, the steps that is followed for providing the provenance graph for the alpha refraction. So this is the process that is followed by the model. Now I'm going to be talking about the alpha diversity box plots with the basic comparison and analysis that uh, have to do. So the data is represented in the form of box plots for item types and persons, and there is a direct comparison for the IDs for the samples, and what was sampled and that was used for the box plot for x and y axis. So this is for the item type, and this is for the person, and this is the visual representation after we did the analysis. So but sometimes it's not clear, and we always have to find the p-values for it, and that is also provided by Chime. So as we can see, for item type the p-value is 2.6, and for person the p-value is 2.6, which is the p-value is 0.25 for item type, and the p-value of person is 0.25, which is not that significant, so we can reject the null hypothesis. And again, the same thing is done for biological sex and ethnicities with the box plot and the p-values, and you can see that the p-values for biological sex is only 0.17 as compared to ethnicity is 0.25, which is greater, I understand, but again the p-value is very small to be significant for a hypothesis testing. Now this is the Peronorma result item type table. It shows the method that I've used, the sample size, number of groups, and p-values. That's just a summary of the method. Now after the analysis, the item types and all of the results that we got was visually represented here. So this is a visual diversity matrix representation of the analysis, as you can see. The main thing of this assignment was to check the significance of the diversity matrix. So on the left we have the box plot for the distance with the groups of distance with the item types and their box plots. And on the right we have the results from the statistical model that method that we were using. That's the results and the p-values associated with it. Again, this is not significant. As you can see, the point, it is 0.3 the p-value for it, so it's not significant. And again, it has been done for all of the samples. So like for example, this is for classes, for persons, and then again for their objects. For this one also the p-value is 0.4 again, which is not significant, so we can reject the null hypothesis. This is for different people who are sampled. There are only 3 because of the histogram that you see before. Anyways, so hang on. Okay, so these are for the objects, and this is for the people. And again, this is the background of what the steps was followed, how you got the data, then it was sequenced and aligned, and then the matrix was formed, and the results were produced. Well, this was my assignment. Thank you very much.